This is my disclosure for the companies that I usually work. Now, this is the topics that I'm going to talk today to make it easy about genomics. I know that this is not an easy topic. We'll talk about some definitions. I will give you an example of how research has been conducted on genes until today, and what is the revolution of omics, and what will represent probably this kind of investigation for the future. So, genetics is what we know, the study of individual genes and their role in inheritance, and genomics, it's usually the collective characterization of these genes and how they represent, together with other enzymes and messenger molecules, how they work. And finally, we have the functional genomics, what represents what is the meaning of this. These groups, and a specific group of genes, what represents in concepts such as biological processes or pathways, including the expression of proteins. You can see here an history of this, from the Gregory Mendel with the inheritance rows to the human genome. You can see here a number of names and Nobel Prizes that they have received for all these genomics and genes. They have, all of them have something in common. They are all men. You cannot see here a single woman. And I want to show you that sometimes there are women that are very important in these steps, but they were not considered. That is the case of Rosalind Franklin, which famous photo 51 about the double helix of DNA was the origin of the famous paper that Crick and Watson published without an initial recognition on that. He never could take the Nobel Prize because he died before the Nobel Prize was awarded in 1962. But I think women like Rosalind Franklin, there are many in science, and usually very much forgotten. Say the next one. This is the two photos of the Nature and Science, where the two kinds of human genome that were developed in the NIH and by Cellular Genomics were published with 1 day of difference, and that is the basis of genomics today. But genomics is not the only concept. This is what usually these sciences study: DNA, proteomics, metabolites, RNA, epigenetic modifications. This is what we obtain from that: genome, proteome, metabolome, transcriptome, and this is the science that tries to explain all of this: genomics, proteomics, metabolomics, transcriptomics, epigenomics, etc., and many other sciences that will appear in the future. For example, the kind of the sense of smell. We have the whole genome with 20,000 genes, the olfactory genome with more than 800 genes, the olfactory proteome with 400 receptors or proteins, and then we have the olfactory function that is much more complicated. In inflammatory mediators, that even comes much more complicated because there are several families that are expressed and several functions. And to join all these concepts are very complicated. We are trying today to link them by endotyping. You can see this paper recently published with different phenotypes or subphenotypes of chronic rhinosinusitis with nasal polyps, in cystic fibrosis, in allergy, in asthma, in eosinophilic fungal sinusitis. And you can see these differences in the expression of different genes. What actually, we don't know much what it means at the end, most of this data. Or the data that Klaus or Seis have shown before, the link of these endotypes, the expression of genes or proteins with phenotypes, asthma comorbidity, polyps, presence of polyps, recurrent disease, that we're trying to link to know what is the meaning of all this data that we're obtaining. To give you an example, we have been studying with aspirin-sensitive patients for many years and trying to figure out what happened in these patients. we end up knowing that many molecules are modified. They have differently expressed, like COX-2, PGE2, the receptor of EP2, etc. At the beginning, we found that COX-2 was down-expressed in epithelial cells. Then other studies that cystic leukotrienes were up and PGE2 were down. There was an imbalance between leukotrienes and prostaglandins. But this doesn't happen in all nasal polyps. For example, in polyps from cystic fibrosis, both enzymes are up in both protein and gene, in COX-1 and COX-2. It doesn't happen the same as in aspirin sensitive. When we stimulate with interleukin-1, also we see that in polyps there is a down regulation no matter where we measure that, in ELISA, in Western blot, in immunofluorescence. But when we measure the own receptor of this interleukin-1, it's also down expressed in these patients. And furthermore, even the EP2 protein receptor, it's down expressed. If we are able to do transfection of this receptor to these cells, we recover all the functions, and all the functions work. What is the final conclusion of all this work that we have done during 20 years? That there are many genes involved in any function. And that is much more complicated than we think. There is downregulation of EP2, there is bad work of CAMP, downregulation of the receptor of interleukin-1, COX-2, and prostaglandins. And if you solve the original, that finally is difficult to find which one is, we can solve the bad cycle that it's induced. That is what is important, that in the future we work with much more data. But this is not easy. That is the resolution of omics that we try to do. We have used a different model. We obtain progenitor stem epithelial cells, and from a single line, we are able to build a whole epithelium differentiated with 3 different cells. Epithelial cells which have ciliated, secrete of mucus, and provide the other origin, the stem cells. Here, in the last one, you can see in green the expression of ciliated cells, and in red the mucus cells. And everything comes from a single type of cells. We are able to build that, and this is important for future cell therapy, that this is very important. But what happens if we measure genes? That becomes much more important, much more complicated. After 28 days of building this epithelium obtained from a single cell, there is a upregulation of genes and downregulation of genes that is different in nasal polyps than in nasal mucosa. You can see here the average. Some in the middle expression of genes are common, going up, and some common, going down. But there are many other genes that are going up or down, and they express in a different way in nasal polyps than in nasal mucosa. But this is only something that we observe. We need to know what this doesn't mean. That is what is called functional enrichment. This data is usually compared or put in set analysis with other data that is already in programs, for example, in the program David. And we link our data with others. And we know what this doesn't mean, all these different expressions. And we obtain this. For example, in nasal mucosa, at day 28, there is a downregulation of many functions, mainly of them derived to cell function, and upregulation on other functions such as the cilium movement or the cilium morphogenesis, which is quite different what happened in nasal polyps. But our interest is compare healthy with pathologic. And what we can observe, that a number of function of nasal polyps with a number of genes, each bubble of this means a conglomerate of genes with a specific function, are downregulated, and most of them related to the cilium movement or the motile cilia assembly, and many others. We know by other studies that the cilia has been studied and has been found problems in many studies, but looking only at very few genes. What we wanted to do is to look at a number of genes. And we see this link between functions in different colors of cilia-related pathways with genes, either if they are upregulated or down-regulated. These are the functions. I'm not going to talk about this. It's only to show you the diversity of analysis that we can do. This is what happened in polyps. You can see that the functions here are much lower because many of them are down-regulated and the number of genes are different. But when we compare these two models, healthy with pathologic, we can see a diversity of expression of different pathways and different processes, biological processes, with a number of genes mainly down-regulated in nasal polyps. And that's not all. I'm going to skip this one. We have done all these pathways, biological processes, looking at mucociliary differentiation of stem cells. But this is regulated by genes, but there are other molecules, such as microRNAs, that are small strands of RNA from non-encoding DNA, that genes that don't express proteins, but these molecules, microRNA, that regulate many genes. And probably this is the future of many treatments. Even the possibility to modify these cells, to convert diseased cells to healthy cells and work with them in cell therapy. That is one of the potential possibilities in the future to cure the disease, in this case, chronic rhinosinusitis. I'm going to show you only, and read it, the last sentence, that is the understanding of new genomic, epigenomic, transcriptomic, proteomics, all the omics that you can rely on, may help precision medicine in using a more personalized, better prediction of success, disease prevention, and patients' participations in the cure, not only in the control, but in the cure of this disease. Thank you very much.